Thank you, Andrew. Good morning, everyone. As Andrew mentioned just now, the research topic of my research initiative project is norovirus aptasensor, which is a new aptamer-based biosensor, and we carried out proof of concept studies to prototype this sensor. Norovirus is the major cause of foodborne and waterborne acute gastroenteritis with various symptoms, often reported as stomach flu. This virus is also known as an environmental contaminant. In fact, it is included in the US EPA contaminant candidate list, which is a list of emerging contaminants known to occur in public water systems. Norovirus has been found at few points in Singapore's urban water cycle. Specifically, it was found to occur in untreated sewage with a concentration of up to 10 to the 6.5 virus per liter, and in treated sewage and surface water, up to 10 to the 5 virus per liter, according to the published reports from Singapore. These results have been obtained using quantitative polymerase chain reaction, which is most widely used for environmental pathogen detection. But it takes at least several hours to obtain the results and has limited on-site application. Whereas biosensor, which rapidly converts a biological response into a measurable signal, potentially enables real-time online monitoring. In this study, we used aptamer as a recognition element and electrochemistry as a means of transduction. Aptamer is a functional DNA or RNA that falls into three-dimensional conformations binding to a specific target and offers various advantages as biorecognition element of biosensor. For electrochemical measurement, we used a standard benchtop workstation as well as a portable measurement system called WeStat, and compared them side by side in terms of their performance as an electrochemical measurement system for our aptasensor using MEMS device. The WeStat is as small as credit card and provided as open source instrument and software with reasonable price, only $130 US. In order to prepare MEMS aptasensor device, we immobilized thiolated aptamer on the MEMS gold electrode through the affinity between thiol and gold. These graphs show cyclic voltammetry profiles before and after immobilization. As you can see here, aptamer immobilization substantially reduced the peak current, indicating that self-assembled aptamer monolayer was formed on the gold electrode. The WeStat showed similar electrochemical profile as the standard benchtop workstation. To further confirm aptamer immobilization on the electrode surface, we labeled the aptamer with a fluorescent dye, FAM, and observed under a laser scanning fluorescent confocal microscope. This image is the top view showing fluorescent signal on the gold electrode. No fluorescent signal was observed on the electrode without aptamer. In this three-dimensional figure, Z-axis represents fluorescent intensity. Fluorescent signal was observed, fluorescent signal derived from the label aptamer was observed only on the gold electrode, as we expected. Now I'm going to show you the results of the detection of virus by aptasensor. We used another electrochemical technique called square wave voltammetry. Square wave voltammetry profile for buffer without virus is shown here. When murine norovirus, which is a model of human norovirus was spiked in this buffer to obtain a final concentration of approximately 50 plaque-forming units per ml. We observed a significant decrease in the peak current. Again, the portable VStat showed square wave voltammetry profile similar to the standard benchtop workstation. Next, in order to assess the sensor's ability for quantification, we made a serial dilution of murine norovirus, spiked in this buffer, and performed square wave voltammetry independently for each virus titer. This graph shows square wave voltammetry profiles, which demonstrates that peak current decreased concentration-dependent manner. By plotting the peak current relative to the baseline against the virus concentration spiked into the buffer, we could draw a standard curve demonstrating a good correlation between virus concentration and peak current. Here I summarize the key progress of this project. First, we developed a novel MEMS aptasensor device modified with aptamer. We confirmed aptamer immobilization with electrochemical measurement as well as visualization of labeled aptamer on the electrode surface. Second, we evaluated a mini electrochemical measurement device, VStat, and observed that it shows similar electrochemical response to the standard benchtop workstation. By combining the MEMS aptamer device, VStat, and Raspberry Pi, we can potentially fabricate a portable and handheld aptasensor platform. Lastly, our experiments have demonstrated rapid and clear concentration-dependent response to virus, which demonstrates its ability for quantification. Deliverables from this project are listed here. Our research paper on this work was just accepted for publication in a peer-reviewed journal. We submitted a patent application, and we presented our work at a few conferences on biosensors. Lastly, I would like to acknowledge this Sensum RI project and our research team from Sensum and NTU. This was a great opportunity to initiate a new research project. Thank you very much, and thank you for listening.
Time for a question, yes.
Hi, thanks for the wonderful speech. I just wanted to know, have you tested this sensor for the real waters, basically for the matrix effect? Because there are many things in the real waters which might hinder the aptamer binding to the sensor. Thank you.
Very good question. The quick answer is we haven't tested real water samples yet, but as you mentioned, there may be some interference substances in the water samples that may inhibit our detection process. So we need to assess the inhibitory effect, and we need to consider some method to purify virus particles or to reduce the inhibition effect, if any. Thank you very much.